All right, it's 11 o'clock. Good morning again. Welcome to the HIV Grand Rounds. I know folks are going to trickle in through the hours, so we will get started so we can have enough time for the presentation. My name is Damilola Jolayemi, and I'm with the UCLA Center for HIV Identification, Prevention, and Treatment Services. And the HIV Grand Rounds today is a great presentation that we're looking forward to. I just wanted to give a brief overview quickly before we get started. Our HIV Grand Round series is offered by UCLA CHIPTS and UCLA Charles Drew CFAR. And it consists of an hour-long lunchtime lecture series that are delivered by invited guests and distinguished members of faculty on a broad range of HIV-related topics. The aims of the Grand Rounds are to share the latest research findings on HIV prevention, care, and treatment, and highlight important developments in HIV-related research, to encourage scientific collaborations and networking between investigators and invited speakers, and to excite an interest in HIV/AIDS research for early career investigators, and also provide resources about new research findings, funding, and career opportunities. So today we will be going ahead and getting started with our Grand Rounds. I just wanted to share that if you have questions, comments, please feel free to use the chat or raise your hand to ask questions. We will be unmuting folks, or allowing folks to unmute to ask their questions. So please feel free to either use the chat or raise your virtual hand. All right, so without further ado, I'm going to go ahead and welcome our moderator for today's HIV Grand Rounds, Dr. Jerome Zach, who is one of the directors at the UCLA Charles Drew CFAR.
Thank you. Good morning, everybody. It's my pleasure today to introduce Dr. Mehdi Bouhaddou, who is a new faculty member in the Department of Microbiology, Immunology, and Molecular Genetics. Just as a way, means of background, Mehdi got his bachelor's at UC Berkeley. He did a PhD at Icahn School of Medicine, and recently completed postdoctoral fellowship at UCSF under Nevin Krogan. He's done some beautiful work on proteomics of virally infected cells, and he's worked with several different types of viruses, but I believe today he's going to focus on SARS coronavirus experiments. So we look forward to introducing him to the AIDS research community here. I think he's going to be a great addition already is a great addition to our faculty, and I look forward to his talk. Mehdi, go for it.
Thank you, Jerry, for the kind introduction. It's great to be here today. Very interested in HIV research. So very, very excited for any potential collaborations that could come out of this and working more with the AIDS community here at UCLA. Can you guys see my slides?
Yes.
Yeah.
Great. Okay, so yeah, as Jerry mentioned, I just arrived at UCLA 2 months ago, so started in February 2023. I'm located in Boyer Hall. I have a co-affiliation with the Institute for Quantitative and Computational Biosciences because my lab is half wet and half dry. So we do a lot of mass spectrometry proteomics and virology, but we also have an arm focused on bioinformatics and mathematical modeling. So if you or any of your trainees are interested in a position, please have them reach out because I'm definitely hiring at the moment. So yeah, as Jerry also mentioned, my postdoc was with Nevin Krogan at UCSF, and I actually went to Nevin's lab to study cancer, but then the COVID pandemic hit, and decided to focus all of my efforts on studying SARS-CoV-2. Had a few other projects in virology in the lab as well, but really wanted to focus on SARS-CoV-2 due to the urgency of the situation at the time. So this was early 2020, and one of the first things we did, just to give you a summary of that background, is we created the first SARS-CoV-2 human protein-protein interaction map. And so to do this, we cloned all of the different open reading frames from the SARS-CoV-2 genome into constructs and ectopically overexpressed them in human cells, performed an affinity purification and mass spectrometry to identify the human proteins that physically interact with the viral proteins. And from this, you can learn a lot about which protein complexes are hijacked by the virus, which pathways are being hijacked, and it allowed us to learn more about how this virus is actually hijacking our cells. Another study that I was intimately involved with was the phosphoproteomics studies, this time we used live virus and simply compared mock-infected cells to SARS-CoV-2 infected cells and harvested lysates at various time points post-infection, and then performed global phosphoproteomics and abundance proteomics. And this is really where my primary interest lies now, and where a lot of my lab will be focusing on is these phosphoproteomics time courses, understanding how viruses hijack phosphorylation signaling in human cells. And then, from these large data sets that we acquire, we can estimate, kinase activities from these profiles based on known kinase substrate relationships. Here are some results from that paper, which allowed us to identify pathways that were being either activated or inhibited during viral infection, and then mapped these, dysregulated kinases to kinase inhibitors, many of which we found to possess antiviral activity. And then the final study that was really, a big part of that 2020 year was now comparing, the same sort of virus host protein interaction networks, but comparing them between SARS-CoV-2, SARS-CoV-1, and MERS-CoV. And this project was incredibly collaborative. It involved hundreds of scientists. I think the paper had 200 authors on it, but hundreds of scientists worldwide really working intimately together to combine different data from different sources. So we primarily worked on the protein-protein interaction work, but others looked at localization of these viral proteins, functional genetics, where we knocked out some of these host factors to look at the impact on viral replication. There was a team sort of focused on structure, and then worked with some clinical data as well. So, these projects were just incredibly collaborative, and it was a real pleasure to be able to work with these scientists at the time. So my general approach to science is to collect a lot of data and then try to make sense of it. So these are the types of data that I typically collect. Some based on nucleic acids like RNA-seq and sort of genomics sequencing, which a lot of the genomics we acquire from public repositories. The RNA-seq we usually send out to get processed. But then all of the proteomics we do in-house. And really, once you collect all this data, I think the question is, how do you make sense of it, right? So that's where I think computational modeling comes in to sort of tie these different data sets together into a cohesive story. And I would argue this is probably the hardest part of the process, because there's a lot of different stories to chase. It's difficult to sometimes interpret what the data means. So typically, once we have some hypotheses, we can then, create more targeted experiments and hypotheses, using functional assays or genetics or pharmacology. But what I've, grown, recently, aware of is that you can actually reinterrogate these same data sets with different questions, right? So you may do a preliminary analysis, which leads you to one hypothesis, and then you could reinterrogate the data set looking at a different subset of genes that may be of interest. So I think there's a lot of richness here that, can be interrogated and reinterrogated. So now I'm going to dive into sort of the meat of the talk today, and focus on one example from recent work. This is work that I was doing towards the end of my postdoc, focusing now on the SARS-CoV-2 variants of concern, and on some of the work that I want to bring into my new lab now. So as all of you are aware of, there were several variants of SARS-CoV-2 variants of concern that circulated around the world. These are known as alpha, beta, gamma, delta, and Omicron. These were predominantly dominated by alpha, delta, and Omicron. And there are various mutations, right, that have accrued within these variants, and most of those mutations are in the spike protein, right, which can affect the affinity of spike for entry receptors on host cells, such as ACE2. They can also impact adaptive evasion from the adaptive immune system. However, the other mutations outside of spike, I think, have been less well studied. I think there's a lot of opportunity to learn about how individual mutations can impact the entire viral life cycle. So that was really our interest especially because our technology was more amenable to understanding what happens when the virus is already inside of the cell. So, this is the basic, the way that the study was set up. So the goal is just understand how these VOC, variant of concern mutations, altered the virus host molecular landscape. And we essentially threw the kitchen sink at this problem. So we designed very tightly controlled experiments this is in collaboration with Greg Towers' group and Claire Jolly at UCL in London. They did the infection experiments. And they were very careful about trying to dose all of these viruses the exact same amount. We have the 5 variants of concern here. This is Omicron BA1. And then we compared those to 2 wave 1 isolates. Vic was isolated from Victoria, Australia, very early in the pandemic. It's thought to be almost identical to the original strain from Wuhan. And then IC19 has the D614G mutation in spike. This was isolated in Europe later in 2020, in the spring of 2020. But essentially took these viruses, infected Calu-3 cells, harvested lysates at 2 time points post-infection, did RNA-seq, abundance proteomics, and phosphoproteomics. We also ectopically overexpressed all of the variant forms of SARS-CoV-2 proteins in HEK293 T cells and performed affinity purification mass spec. And there are 3 major strategies that we noticed that these variants were using to during the course of their evolution. So we noticed that all of the variants altered their viral gene expression during infection relative to wave 1 viruses. We also noticed that all of the variants had some evidence of altered viral protein phosphorylation, specifically phosphorylation of the nucleocapsid protein. And then, actually I should say all but one, all but gamma. And then all of them altered virus host protein interactions in some way through those protein coding mutations that appeared in the genome. So today I'm going to really focus on the later part of my talk, talking about how we can combine these different observations and one particular story related to innate immune evasion by the variants of concern. So here is some plots of viral replication over time, the qPCR, the real-time PCR on the left, and the RNA-seq on the right. All of these experiments were run side by side for the PCR and the RNA-seq and the proteomics. Everything was done at the exact same time, but it was done in 2 different experimental batches. Each batch has 3 biological replicates of each virus at each time point. And so we did that to sort of have, there's some overlap between experiment 1 and experiment 2, but not perfect. Overall, we see replication is similar between the viruses. There's definitely some differences that become apparent more in the RNA-seq data, which is on a linear scale. But we definitely noticed that Omicron was replicating more slowly than the other viruses, given the same input dose of the virus. So we'll come back to what that can mean later in the talk. If we just look at the fraction of detected molecules that change significantly at any time point in any virus, we're seeing that's sort of the black in these bars. We're seeing most of the changes occur at the level of RNA and phosphorylation, which you might expect given the short time course of 24 hours. Probably protein abundance changes would occur more later. And then this is just a breakdown of the directionality of these changes at the RNA, protein, and phosphorylation level over time. So going back to the SARS-CoV-2 genome, there's 2 main parts of the genome that, or how I think of it, is there's sort of a constant region and a subgenomic region. So when the virus enters the cell, it will eject its single-stranded RNA genome into the cell, which can then be translated by the host ribosome to create the replication complex, which can replicate the single-stranded RNA genome. But then it also produces these subgenomic RNAs, which code for the structural and accessory gene proteins of the virus. And those are produced separately in these double membrane vesicles here that then eventually get produced into protein and then form the new viral particle. Interestingly, when you compare the genomes of different coronavirus species, you see that this constant region is fairly conserved between different coronaviruses. And really the major differences are in the accessory proteins. So which accessory proteins are present and what sequence they have, which really suggests that these accessory proteins are what drives adaptation of the virus to new hosts or adaptation to different stressors in the environment. So this sort of theme was really interesting to us at the time. And when we look now at viral protein expression, we actually do see changes in this same sort of subgenomic region of structural and accessory proteins. So what I'm showing here now is viral RNA levels, and the normalization here is really important. So here we're normalizing each variant to the amount of genomic reads. So that's the amount of ORF1ab reads that have that leader sequence at the beginning of it. And that's to normalize for any differences in the amount of virus that may have ended up entering those cells. And then we're also normalizing to VIC within the viral protein. So everything is relative to VIC, which is our early lineage control. So if it goes up to the right, that means it's more than the early lineage, and then to the left, less. So we definitely see a lot of these structural and accessory proteins changing in abundance at the RNA level, and then also at the protein level. So we don't see as many changes in the non-structural proteins, but we do see much more robust changes for structural and accessory proteins. And what's interesting is that all of the variants of concern upregulated at least one viral protein by twofold, right? So it seems like across the board, they're all using this strategy to some extent during the course of increasing their fitness, right? So this suggests that maybe this does impact the growing fitness of these variants over time. So as you may have noticed from the last slide, there are some instances where the regulation of transcription and translation are disjointed. So more RNA does not necessarily always mean more protein. So this is one example. Alpha and Delta have very different levels of ORF9b RNA, but have about the same levels of ORF9b protein. So ORF9b is actually a gene that's within the nucleocapsid segment of the genome. It's actually out of frame. And when you compare the sequences from these different variants at this region, so here we have the start of N here, this ATG, and then a few base pairs later, you have the start of ORF9b. And we noticed that for alpha, there's two mutations in this region. There's one called what we call the -3 deletion, which is a deleted nucleic acid 3 base pairs upstream of this N star site. And then it also has this D3L mutation where the GAT is changed to a CTA. And this mutation probably has a lot of different impacts on the virus. But then, so looking at Delta, we found that there's only this -3 deletion and not this corresponding CTA mutation. So our hypothesis was that, and this was also hypothesized by others at the time, we were not the only ones, was that the CTA mutation, or CUA, I guess I should say, acts to enhance the transcriptional regulatory sequence of that region, which would essentially increase the amount of ORF9b subgenomic RNA that's being read or produced from the genome of the virus. However, this -3 deletion would impact leaky scanning of the ribosome. So in normal cases, without that -3 deletion, the host ribosome would translate N protein from the N subgenomic RNA. However, with that -3 deletion, the ribosome might scan. The Kozak sequence is not as strong, right? It's sort of predicted to be a weaker Kozak consensus sequence, which would then cause the ribosome to scan and start translating more ORF9b. So in collaboration with Luis Martinez and Cheng Yi at the Texas Biomedical Research Institute, they're sort of masters at creating these mutant viruses. They actually were able to create 3 different viruses, one with just the D3L mutation, one with the -3 deletion, and then one with both of the mutations. And this is all in the sort of Wuhan background strain. And what they found was exactly what we expected, that the D3L would produce RNA, but not necessarily protein, also speaking to the fact that ORF9b subgenomic RNA doesn't seem to lead to a lot of protein produced, but most of the protein for ORF9b seems to come from the N subgenomic RNA. But then if they had just the -3 deletion, they had not as much RNA, but had a lot more protein. And then if you have both of the mutations, you can get both RNA and protein. And this corresponds perfectly to what we're seeing in the actual sequences from the variants of concern, and really helps to explain why alpha and delta have different levels of ORF9b RNA, but the same levels of ORF9b protein. So, what does ORF9b do, right? So ORF9b, one of the interactions we were very interested in early on, this is from our first paper looking at the virus host protein interaction map, is an interaction between ORF9b and Tom70. There was actually a crystal structure that was solved primarily by Klim Verba at UCSF, who found ORF9b to bind within a very conserved pocket within Tom70, and the interaction seemed to be very tight, based on the proteomics data. So our hypothesis at the time was that Tom70 is known to associate with MAVs, and that complex can activate TBK1 and IRF3 to stimulate the interferon, production of interferon. And so we hypothesized that ORF9b could be coming in here to antagonize this process and essentially act as an innate immune antagonist protein. It sort of as a side note, we also noticed certain phosphorylation sites on ORF9b at serine 50 and 53, which in interrogating the structure, we found that these are actually specifically the serine 53 seemed to be important for the interaction between ORF9b and Tom70. So it could have been that, or we proposed that the phosphorylation of serine 53 might disrupt this interaction. And indeed, if you make a phosphomimetic, this is work by Jyoti Batra, because I am terrible at Western blots. I could never make a Western blot this nice. Jyoti Batra at UCSF, what she did is she created this mutant with serine 53 changed to a glutamic acid, which is supposed to be mimic a phosphorylation site, act as a phosphomimetic. And if you here she's overexpressing ORF9b and with a strep, and then Tom70 with the flag, and she finds the interaction to be lost, essentially, right? When you have that serine 53 changed to glutamic acid, the interaction is lost with Tom70, indicating that phosphorylation of that site really could potentially disrupt that interaction. And then the other assay that we did is here we're stimulating HEC293 T cells with an IHG reporter with different amounts of poly-IC, and you can see that co-transfection of ORF9b wild type does is able to suppress that response and act as an antagonist of the innate immune activation relative to empty vector. But if you have this mutant with these phosphomimetics, you're not able to antagonize the innate immune response, indicating that interaction with Tom70 is actually critical to the innate immune antagonist function of ORF9b. So then, my interest is really in kinase, phosphorylation signaling. So my immediate question, well, what kinase is doing this? What kinase is phosphorylating this serine 53 residue? And, what was very surprising at the time was that TBK1 really came up as the top hit based on this computational prediction tool. So this is in collaboration with a postdoc that was working with me in the lab, Jack Moen. We bought some TBK kinase and ordered ORF9b peptides with, either ones that were, of wave 1 origin or with the serine 53 change to an alanine. And we found that indeed, if you change that serine 53 to an alanine, you no longer get sort of phosphorylation by TBK1, sort of indicating that TBK1 kinase is responsible for, or can at least phosphorylate that site. So this set up a really strange situation where the ORF9b is coming in to antagonize innate immune activation, essentially antagonize TBK1 activity, but then TBK1 can feed back to de-repress this inhibition, right? By phosphorylating, ORF9b stops prevents ORF9b from being able to antagonize it, which sets up this sort of double negative loop, right? So ORF9b can inhibit TBK1 and TBK1 can inhibit ORF9b, which certainly seems perplexing, right? So what are the dynamics of that, right? Is it sort of an early infection versus late infection switch? You know, maybe early in infection, ORF9b is not phosphorylated, but later it is. So I think, you know, one of the projects in my lab is really trying to understand what is the dynamical interplay between ORF9b expression during the time course of viral infection and then TBK1 activity, and when does that sort of turn on, and when does that phosphorylation event of ORF9b become the strongest during viral infection? So I'm not going to go into this in much detail. I just wanted to present it quickly. You know, this is now going back to the variants of concern. We essentially, as I mentioned, profiled every single mutated VOC protein and noticed that you know, all of the VOCs had some evidence of changes, right, in response to these protein coding mutations in virus host protein interactions. And we have this beautiful map that is unfortunately in the supplementary figure for our paper because the reviewers didn't like it. But I think there's a lot of richness in here. And so, you know, red line means the interaction is increasing and the blue line means the interaction is decreasing in that mutant, right? Like you have NSP13 delta, so that would be the mutant compared to the wave 1 version of that protein. So I think if you are interested in virus host protein interactions in SARS-CoV-2, I encourage you to look at this map so that it gets some use. So yeah, I think now that we've collected all this data, there was a question, you know, how do you make sense of this? And this is where the computational modeling comes in. And we actually decided to do something relatively, you know, simple. We didn't want to do something that was too complex. So what we did was we took all of the genes that were significantly changing during infection across these different data types, right? So this is like the number of number of RNA genes that change. You know, this is irrespective of which variant. This is just all the genes that have some evidence of change, right, during the course of infection. And we performed a network sort of modeling approach where we essentially took all of these genes and overlaid them onto the string network, and then used the string network to define modules of sort of different pathways that were interconnected within the network and also regulated during infection. And this allowed us to define 85 different pathway modules. And to do this for the computational people, we used a diffusion state distance metric combined with a hierarchical clustering algorithm to sort of create these different modules. And the first thing we did was actually just to look, okay, let's plot all the modules across all the variants, and let's see what that looks like. So in the gray lines, you have each line is a different module. And what we noticed is that they're all sort of following the same trend, right? Like if you see an increase in, this is also, I should mention, the absolute value log 2 fold change in that virus, right, of all the genes in that module, right? So if it goes up, that means that there's more changes happening in that module. But they seem to all go up together to the point where we could even define sort of an average host response, right? And you know, probably some of you have guessed that this correlates very well with the amount of virus in the cells, right? Which would make sense. If there's more virus in the cell, there might be more of a there should be more of a host response to that virus. And we definitely see that this average host response correlates very well with the amount of viral protein that's produced in the cell. And so we decided to see if we could use this to our benefit, because one issue is that, you know, if we were to say that Delta had more of an impact on the host, that would be misleading, because it could be that Delta just has more, the replication for Delta is higher. So we decided to look at a correlation between each of these pathway modules and the average host response. And we reasoned that if this correlation was strong, then pathway regulation was likely similar across the variants, and that it was simply a factor of how much virus there was, right? Which was probably less interesting because it didn't really highlight individual differences, but more just it would highlight that having more virus would elicit more changes. However, if the correlation between the passive pathway module and the AHR was weak, or maybe even negatively correlated, that might, or just maybe I should say not correlated, that would suggest that the variants are differentially regulating this pathway through some sort of mechanistic, path, some sort of mechanism is specific to a variant, and not directly related to how much virus is in the dish. And if we were to label each of these categories, one would be really good potentially for finding pan-variant antiviral targets, right? If you can find pathways that are similarly regulated across the variants, those might be good targets. And then for the different pathways, that would be probably better for understanding mechanisms of pathogenesis. So we did some analysis. We again combined across data sets to just define what are the 10 most correlated modules with AHR and what are the 10 least correlated modules. So the top 10 things that really jumped out to us were things like cell cycle, translation, and this makes sense because all the variants need to hijack translation, right, during the course of their infection. But interestingly, in the very bottom of this, the least correlated were related to the innate immune response, which really suggested that the variants were doing something to all, like they weren't all hijacking or modulating the innate immune response similarly, that there may be differences between them. So in the spirit of the lab, we then reinterrogated the same data sets with a new question. And we found a set of 61 ISGs from a recent publication. And we essentially looked at what is the average expression of these ISGs across all the variants in our data sets. And what we found is that all of so alpha, beta, gamma, and delta seem to be more strongly antagonizing ISG production relative to Vic, whereas Omicron was doing the opposite. It was really triggering ISG production, and a Western blot confirmation of this, looking at specific genes, you can really see that decreased expression of these ISGs for alpha through delta, but then an increase for Omicron. And I want to mention that this was irrespective of how much virus was in the dish. I should say that Omicron had lower replication, right? So this is over time, 10 hours, 24 hours. The x-axis is how much virus, how many genomic counts for each virus, and we do see that Omicron has less. So despite having less virus in the dish, there was actually more ISG induction. Omicron is also still sensitive to the interferon that it produces. So this is an experiment where you treat the cells with ruxolitinib, which is a JAK-STAT inhibitor. You can think of it as sort of shutting off interferon signaling in your cells. So what we see is a certain amount of rescue, right? Because if you antagonize interferon, the virus should replicate better. But you see a much more massive rescue, a much greater rescue, maybe twofold greater rescue for Omicron than you do for alpha and delta in the presence of ruxolitinib, suggesting that Omicron is, yes, it's producing a lot of interferon, but it remains sensitive to that interferon. It hasn't developed mechanisms to become resistant to that interferon. So, going back to our protein expression data, one thing we mentioned, we sort of observed off the bat is a decreased expression of ORF6. And ORF6 is well known to antagonize the innate immune response. So, in the pathway, you have RNA from the virus, which is sensed in the cytoplasm and results in the phosphorylation of IRF3, as well as others, which then translocate into the nucleus. Well, ORF6 is known to bind to the nuclear pore complex and antagonize, to block the ability of IRF3 to enter into the nucleus. It can also, later on block the translocation of STAT1 and STAT2 into the nucleus, as well, which is not shown here. Sort of recent studies have shown that it can also block the export of ISG RNA back into the cytoplasm. So it's a very potent innate immune antagonist. And this is in collaboration with Massimo Palmerini at the University of Glasgow, who was able to generate an alpha virus that had ORF6 knocked out. So if you compare IHG antagonism of alpha compared to one without ORF6, you definitely see like a much a switch in IHG production in response to this virus. So we then, at the same time, we were trying to wrap up this paper, but then the Omicron subvariants were emerging. And so we decided to do one more experiment where we would compare, BA1 to the emerging subvariants BA245. And luckily we've stopped now. We're trying to wrap this up once and for all. But what we noticed, which was really interesting, is again, looking at IHG expression, this is now all relative to BA1 in this experiment. We saw that there was sort of this, evolution of enhanced innate immune antagonism by BA5. So although BA1 wasn't antagonizing, as I showed you before, BA5 was starting to antagonize again, which was interesting. And, looking at the viral protein expression levels from this experiment, we actually do see that ORF6, was able, these viruses, encoded mutations that upregulated ORF6, which could explain why you see an enhancement of innate immune antagonism. You might have noticed that, BA5 is really the strongest antagonist, relative to BA4, but they have actually the same levels of ORF6 upregulation. So that sort of puzzled us, right? If they have the same amount of ORF6, maybe something different is happening for BA4 rather than BA5. And looking back at the mutations, we actually noticed that there's a mutation in ORF6 in BA4, which is also in BA2. This is known as the D61L mutation, and it's a protein coding mutation in ORF6. So we wondered if that could explain why BA4 was not as good at antagonizing IHGs as BA5. So we reinterrogated our APMS data. So we had run this experiment previously, and we noticed that indeed, if you look at the fold change of these host proteins in the AP from the D61L compared to the wave1, we see a decreased affinity for RAY1 and NUP98, which are an integral part of the nuclear pore complex. So, there was actually a published structure of this interaction between ORF6 and RAY1 online. And what we noticed is that this interaction between D61 and RAY1 actually forms a salt bridge, which could, play an important role in stabilizing that interaction. Interestingly, not as much as another amino acid here, M58, which really protrudes into a pocket within RAY1. And if you mutate M58, you really, very strongly disrupt. You can't even detect RAY1 and NUP98 in the affinity purification. But D61 actually, reduces it strongly, but not, completely. So we're interested also in understanding, why this mutation and not another, and sort of, is this sort of tuning the response, or is it just sort of a weaker overall response? So this is a sort of model figure of what I just mentioned. If you have the wave 1 version of over 6, you get sort of strong innate immune antagonism of the innate immune response. However, if you have this D61L point mutation, that decreases the interaction between ORF6 and RAY1 and NUP98, and thus decreases the antagonism, reduces the antagonism of the innate immune response. So, when we were putting this paper together, the editor really wanted a final figure sort of explaining what we found. And so we decided to generate this figure on the left, which is all data driven, and basically shows in green the similarity of spike from between each variant and wave 1, right? So the similarity decreases a bit, right, for alpha, beta, gamma, delta, and then decreases much more strongly for BA1, right? BA1 had 36 mutations in the spike protein. Depends how you define mutations, but and then and then, yeah, so then if you also overlay the IHG expression on top of this, what we notice is that, for alpha, beta, gamma, and delta, you have antagonism of IHGs, which then completely switches in conjunction with a really strong, increase in spike mutations, which then over time, as sort of the Omicron subvariants are evolving, we see a renewed increase in innate immune antagonism, even though the spike is actually not changing as much. And I haven't actually presented this before, so I'd be curious to know what people think. And feel free to shoot me an email or say something after during the Q&A. And we don't know if this is correct or not. We're still playing with this, but there seems to be if there's a very strong adaptive immune escape, right, which was the case for BA1, it was able to very strongly escape the adaptive immune system because it had a very much remodeled spike protein. Then there's less of a need for innate immune suppression, right? So it can still be very successful, infecting a lot of the transmitting at a very high rate, even if it doesn't have as strong of innate immune suppression. And I should mention that Omicron BA1, one of the hypotheses is that it evolved within an immunocompromised patient, which might decrease the pressure to enhance innate immune suppression. But there's maybe this relationship where depending on the amount of adaptive immune escape, you have sort of a decreased or altered need for this innate immune suppression. Okay, so just to quickly wrap up this, so where am I going now, right? Where's my lab going now after this work? So my main interest is really in understanding how viruses exchange with human kinases and human kinase signaling. What does this exchange mean for the virus, for the host, and for developing new antivirals? So I'm working with 3 really talented rotation students in the lab who are, there's this recent paper that came out of Lou Cantley's lab. They really comprehensively defined kinase motifs for the entire serine-threonine kinome. This is this is just in peptide sequences, but they also developed a computational tool where you can now put in your own sequence and it will tell you which kinase is predicted to phosphorylate that sequence. So for this rotation project, what we did is we ran 121 different human infecting viruses through this pipeline to really comprehensively define, based on this computational tool, which human kinases phosphorylate which viruses and to what extent. This is a plot that my student Erin made, and what she did is she essentially took the number of high confidence kinase motifs in each viral proteome and divided it by how many amino acids that virus had in its proteome, right, to normalize for just the amount of, because each virus can have a very different sized proteome. And it definitely seems like certain viruses have more heightened conservation of these kinase motifs than other viruses. Specifically, what we're seeing now is a lot of hepatitis viruses are tending to come up towards the top of this list. So it's an interesting question. Why are these viruses evolving to encode these sort of conserved human kinase motifs? Maybe there's a reason for that during the course of their infection, maybe sort of switch the function of certain viral proteins or otherwise impact the viral life cycle. So we're very interested in following up some of these hits and really looking at, okay, which kinases are we talking about, which pathways, et cetera. And with that, I just want to thank a lot of people who contributed to this work. I think I feel extremely lucky to have been at the right place at the right time. We working on with the Quantitative Biosciences Institute at UCSF with Nevin Krogan and the team has been was extremely really fun, I would say. As I mentioned, Greg Towers and Claire Jolly did a lot of the BSL-3 SARS-CoV-2 work for the initial experiments. Adolfo also helped a lot with the BSL-3 work and has been a very close collaborator for a long time. I actually went to Mount Sinai for my PhD, so I know Adolfo from PhD. And then Luis and Massimo really helped to create these mutant viruses, which is, I think, very tricky science and not something that comes easy. And then Lorena and Danielle really helped on the UCS side with the proteomics and the virology. Pedro really helps a lot with the bioinformatics and sort of mentoring some of the bioinformatic processes that went into this. The work of Jennifer, I didn't show, but she's been really instrumental in creating these viral-like particle systems for SARS-CoV-2 that we looked into, and with her postdoc, Syed, who's been extremely helpful in that process. And then Rodit and Manolis have really helped also with the computational aspects of the work, and Marco with the virology. And with that, I'm happy to take questions. And if you don't want to ask a question, please send me a follow-up email if you see anything that we could work on together. Thanks.
Mehdi, thank you very much. That was beautiful. I think you're going to add an important, great new dimension to the science being done here.
Great.
So I have a question that mostly just reflects my lack of knowledge along these areas, but you talked about all these different variants and different pathogenic processes and how they escape innate immunity. Are these mostly thought to be sequential mutants that are changing, or is it scattered? I don't know how I'm saying this right. Is it scattered regionally? Do you understand my question?
Do you mean like, did one variant come from the other, from the previous variant?
Yeah, the question is how sequential are they, or how independent?
Yeah. So it's my understanding that they all seem to evolve separately. Like they didn't evolve from one another. Alpha, beta, gamma, delta, Omicron sort of were all present at some level sort of early in the pandemic and sort of emerged at different points. That they did not sequentially evolve one from the other. I hope I'm saying that correct, but I'm pretty sure that's correct. However, the Omicron subvariants are thought, I think, to evolve from each other, right?
That makes sense. You did show a couple that had, they each had the same mutation, and then the other one had a different, an additional mutation. So that was sequential, but yeah.
Yeah, exactly, yeah.
Cool. Does anybody else have a question for Mehdi? So I think it would be extremely exciting, Mehdi, if you do move into HIV. There'd be a lot of investigators here that would want to help you out or collaborate with you. I think it'd be really.
I'd love that.
Yeah, I think it'd be really.
Yeah, I'd really love that. I don't know much about HIV, but I'm very interested in it. And if anyone wants to do some proteomics, maybe that's where I could slot in.
Yeah, there are definitely people here that could help you out. Melody, you have a question.
Yes. Hi, Mehdi. That was a great talk. Really enjoyed it. I thought it was so interesting how you plotted the variance progression, with the ISG expression. And I'm just curious, since there's so much sequence information out there, thinking about the Omicron, subvariance divergence, right? Because there's so many of them, and the tree is so extensive. Do you think it would be interesting to actually see if you can identify the sequences of the ones that didn't quite make it, were not as successful, and compare those sequences with the ones that eventually became the major sort of divergence, or the tree, majority of the tree, phylogenetic tree, if that will also give you some insights in terms of, the hypothesis you're trying to test here.
That's
the balancing
super
act between the adaptive and the innate immune suppression.
Yeah, that's a really great idea. I mean, the nice thing about doing this mechanistic molecular virology work is that you can now understand what impact these mutations have, right? So then you can look back at other sequences and sort of compare, oh, well, this one doesn't have that mutation, therefore we predict it wouldn't have ORF9b upregulated, therefore maybe it doesn't antagonize the innate immune response as well, right? And maybe that's why it wasn't as dominant. So yeah, I think that's really interesting. That could be a whole project, right? To sort of take the mutations that we understand and then compare with all these sequences. I think that hasn't really been done as far as I know. And I think that'd be very interesting. Yeah.
Well, thank you.
It'd be great to also isolate some of those viruses and then put them in culture and then do the proteomics to confirm.
I don't know if people have done that yet.
Yeah.
Lulan has a question.
Hi, Mandy. This is really great talk. Thank you so much. My name is Lulan. I work for Ken Hong's lab. I have a quick question for you. As the virus continues to mutate, do you think this is really subjective, I think, but do you think that some of the mutations that you find that are interesting could be applicable to a different strain that carries the same mutation? Or if so, is there computationally, can you determine or predetermine if a new strain that carries a similar mutation could have an sort of like an effect of that is being carried over?
Yeah, you mean if the same mutation will have the same functional impact in different viruses, basically? Is that your question?
Essentially the same virus, but obviously different lineage or different subclades.
Yeah. I mean, look, I think there's a lot of complexity to this. I mean, if a virus always had that -3 deletion, for example, upstream of N star, I would probably assume that yes, it would have more ORF9b, but there could be other factors that impact ORF9b stability or that impact that Kozak sequence. Like maybe there's another mutation in that Kozak sequence that could have a different effect, right? So I think you have to be sort of careful, but in principle, I would say that it's not a bad route to follow, right? If you understand what one mutation does, you could potentially extrapolate its impact to a new virus, as long as you can validate that and test it. I think that would be a good way to generate a hypothesis, right, for a new virus. Yeah, and I think that's the sort of goal behind this work, is if we can understand what certain mutations are doing, maybe we can prepare for future viruses better, right? So if we see that a future variant has these mutations, maybe we can start to build some hypotheses about how it has become more successful, right? And what are the mechanisms underlying that? Did that answer your question?
Yeah, that's right. Thanks. Yeah.
Okay.
Are there other questions for Mehdi out there? Doesn't look like it. In that case, Mehdi, thank you so much for giving us this great lecture. We're excited to see what you do in the future.
Yeah, thanks so much, Jay. That was really fun. Thanks everyone for joining.
Take care, everyone.